The second part of photosynthesis is another set of reactions, and these are sometimes referred to as the dark reactions. They are more accurately called the light-independent reactions, which is also referred to as the carbon fixation cycle or the Calvin cycle. Yes, there are many names. Mainly, people have probably heard the dark reactions or the Calvin cycle, and I'm going to offer one more way to think of this. This set of reactions occurs after the plant has already absorbed the photon of light and used that photon to make some ATP. And in this step, the plant or other organism is going to use that ATP to then make glucose. And so that is our energy storage molecule. And so you can also think of this as the synthesis reactions. Now, hopefully that adds to helping you remember this instead of just being another name for this, because photosynthesis, the first part of it, the light reactions are the photo part. And then the second part is the synthesis, and we're synthesizing glucose. All right, so in the dark reactions or the carbon fixation cycle, etc., this is where the plant is going to use carbon from carbon dioxide in the air to build glucose. And if you know the chemical formula for glucose, you know that it has a number of carbons in it, and those carbons all come from carbon dioxide. So again, the plant is literally taking molecules out of the air to build its glucose. Pretty cool. And definitely different from what we do as animals. So the dark reactions are diagrammed here. This is complicated, it's a little bit intimidating, but let's go through this step by step. You definitely don't need to know all of this for any of my classes. So in the dark reactions, the inputs, as I said, CO2, as well as ATP, which I mentioned a moment ago, and also NADPH. NADPH is from the light reactions or the photo reactions, and you could go back and watch my video on that if you're forgetting or blanking on what NADPH is. And it's just essentially, in this case, an energy carrier molecule. Now, what we're going to get out of the dark reactions or the light independent reactions are the used up form of ATP, which is ADP, used NADP, which is just the NADPH with the H removed. So both of these molecules can be reused in the next set of photo reactions. And then the real thing that we're interested in is the glucose or whatever other carbon-based molecule the cell is in need of at this time. So this is a more simplified version of what we saw just a moment ago, where we're seeing the Calvin cycle just simplified. We don't see all the molecules, but we do see the important inputs and outputs. We have carbon dioxide coming in at the top, and then at the bottom we have sugar coming out, and that covers glucose and other various sugars that you could make with this process or that the plant could make. We can't do it, so you can't do it. And then we also see that there are arrows showing NADPH coming into the Calvin cycle and ATP coming in, and then ADP and NADP leaving. And these are the really important things to know. Now, the one other thing that I want to note is going back to the other slide that we saw a moment ago. It's a little bit more complicated again, and you definitely don't have to know all of this, but what I want to point out is this enzyme that's pointed out in pink here, and it's called Rubisco. That's the shortened or abbreviated version of this enzyme's name. Rubisco is really important because it is in charge of getting those carbon dioxide molecules in a form that can be used in the Calvin cycle or in the dark reactions. And you might say, oh, well, I mean, every enzyme in this process is in charge of something important, right? Yes, but the problem is that Rubisco is not good at its job. It is a really, really large enzyme, and it's also very slow and relatively inaccurate. And that used to be okay millions and millions, or maybe even billions of years ago. Yeah, billions, because there wasn't as much oxygen in the atmosphere. There were higher levels of CO2. So when there was more CO2 and less just O2 to bind to Rubisco and confuse it, then Rubisco was always doing its job just fine. Then when plants put more oxygen, plants and algae and bacteria put more oxygen into our atmosphere, now Rubisco can end up binding oxygen, O2, instead of CO2, and actually that's the reverse of what we want the Rubisco to do, what the plants wants Rubisco to do. And again, that's just because of the higher concentration of O2 in the atmosphere now, as opposed to when Rubisco was first evolved, or you could think of it as invented. And so this does cause problems for different plants, especially plants that live in very hot climates, because then they are going to lose the CO2 and need to bring in more CO2 through their stomata, and water can also escape through stomata. So losing the CO2 in itself is not such a big deal, but in order to get CO2, the plants put themselves at risk of losing water as well. So you can learn more about that in other videos on YouTube or textbooks. But I just wanted to highlight that enzyme because it's really interesting and also problematic for the plants. The other thing I want to mention here that we also saw on the simplified diagram was that this is a cycle, right? And so the final version of the carbon skeleton, we can take it out at the bottom here. We could take sugar out, but we could also allow the carbon skeleton to continue and be recycled to combine with Rubisco. And so we're going to see this in a few in cellular respiration as well when we talk about the Krebs cycle. So you could bring things in, you could use them, and then take them out, siphon them off as you need them, still leaving some carbons in the cycle because they're continuously coming in, but sometimes you could also leave these carbons in so that you are going to replenish the cycle. And here I've just labeled where these molecules come from. So ATP comes from photosystems 1 and 2. NADPH comes from photosystem 1. And then our G3P is just the molecule that is used to then build glucose. But you see that basically you have 3 carbons here, and our glucose is a 6-carbon molecule, so you have a half of the glucose here. You get another one, and you can make glucose. And so I wanted to mention also that plants don't just use glucose for energy storage like we do. They also use glucose to make their cell walls. So glucose can be used as a polymer. There are different types of polymers of glucose, so we use glucose polymers to store our glucose. But plants also, so they have starch, which is a form of glucose polymer for energy storage, but they also have a different polymer of glucose. The glucose molecules are bound together differently, and this is cellulose, which is again an indigestible or molecule. We sometimes also refer to it as fiber, which incorporates a couple of different things, but cellulose is one of them. And again, this is a polymer of glucose. So really when you're eating plants like lettuce or other vegetables, you're consuming a lot of glucose, which potentially could give you a lot of energy, but we cannot break down this polymer, so we don't get a lot of energy from it. And that's why we don't have a lot of calories coming from things like lettuce. And so this is just another view of this glucose polymer, but you can see here how it's you can stack the linear polymers on top of each other and bind them together, so you can get that really strong cell wall. And this is an electron micrograph of cellulose fibers.